Okay. First of all, I want to thank everyone for coming to beautiful Hamburg. I'm especially happy to see all 15 ESR students here. So that's very exciting for my part. Today I will be talking about modeling rare earth elements in the aquatic food web. I've decided to show this to you to also give you a perspective of how we could use ecotoxicological data in a different way instead of the standard bioassays we see. Special thanks to my co-author Jordi, who's from SK CEN in Belgium, and he has been a great help and support for me. And I will walk you slowly through how we go from these kind of experiments to a food web model. Oh, that's not work, of course. So, as you might have understood, is that most studies focus on one species, as is recommended by the International Standard Operation Procedures. Examples of this we have seen from Edith and Marion's talks, but also from what you have done yesterday in the lab. This is not necessarily a bad thing, as it helps us understand how an individual species or an individual organism is impacted by contaminants, such as rare earth elements. But it's also due to the fact that aquatic ecosystems that we find in the nature are very complex. You have a lot of factors that play into it, both chemical, but also biological, temperature, light, whatever you want to say. And this makes studying aquatic ecosystems very time-consuming, also very expensive, and it often also is sometimes impractical to do. As Susanna mentioned, with this River Rhine, you have tides, and when it's high water, you can't basically do anything. So a food web model is a tool that could help us understand and predict natural ecosystems better. A food web model is basically a network of species connected by different trophic feeding links that generally point from predator to prey. So this is the easiest example that we know. Algae, the prey species, gets eaten by Daphnids, the predator species. So the arrow points from prey to predator. This is, however, not how we know food web models. We know food web models more like this, a way more complex situation where you have one prey species that is fed up on by different predators and also feeds up on different prey themselves. However, reality is more like this. I don't think any one of us has a clue of what's going on here. There are a lot of biological and abiotic factors that play into it, but this is reality. This is what we see in nature. So a bit taking a step back, modeling is very interdisciplinary. You need a lot of background to understand this bunch of lines we just saw. You need to have knowledge of chemistry, of biological effects, but also know mathematics and sometimes even computer coding. So in general, when we talk about models, we don't talk about photo models, but we talk about mathematical representations of a system. It's a heuristic tool for guiding further studies. This means that we try to see what aspects of the system are in most need to study further and where more data would be needed. Basically, where are the data gaps? So, also important is that we understand the limitations of models. So even for models, reality is way too complex to capture. And because of this, it requires simplification, so sacrifice of details. However, this leads to uncertainty, but uncertainty is okay, because in order to understand the full model, you would need to know everything, and we don't know everything, so you can model. Also, it has to be kept in mind that it's not a substitute for reality, it's still a tool, but it's still a realistic representation of our natural systems. So my objective with the development of the food web model is to basically develop a simple aquatic food web model to identify the chronic impacts of two rare elements, lanthanum and gallium, on multiple biological species, and then that this model could then be used to apply it in the development of new insights in different laboratory and field conditions. We aim to do this by looking at the knowledge gaps of previous studies but also do extensive literature research and look into databases of which information is readily available, and perform not only individual bioassays, but also experiments related to food web modeling, so-called COSM experiments. I will talk about that a bit more later on. So my current prototype, because it is a prototype, is built in Model Maker 3. This is a software model that allows me to build an open model. Open model means that as soon as one of our ESR has data, I can put it in. As soon as you, as senior researchers, have data, I can put it in. As soon as government gets data available, I can put it in. A downside is a bit that there's a time limit, because the model runs only optimal when there's 14 days considered. So this means that doing the chronic dyspnea experiment, for example, which is 21 days, I can't fully incorporate the data. My current model is based on existing models that are not necessarily related to rare elements, but for example, to copper or just in general prey-predator relationships. It considers, however, environmentally relevant concentrations of rare elements, because I'm still trying to predict the natural system. And it has a sediment water interface for 5 different organisms. As we know that rare elements stick to sediments and fine organic particle matter, but they might also float up into the water. I look at various trophic levels of organisms. And so basically, this is a very simplified version of my model. So this is how you start with your model. You basically get a piece of paper and you draw the easiest interactions you have for your food web. So we start with the primary consumers, which are at the bottom of the food web. So we're talking about microalgae and bacteria, which, as you can see, get eaten by the secondary consumers, which are the dephnids and the nematodes, who in turn gets eaten by the ostracods, which is the third consumer level. As you can see, the rare elements don't only impact the primary consumers, but they also impact the secondary and third consumer levels. So when you do modeling, you require mathematical equations. One of the most mathematical equations used is the Lotka-Volterra equation, which is the principle that when the prey goes up, the predator goes up, and when the prey goes down, the predator goes down. Next to that, I also incorporate first-order linear processes to look at the transfer of rare elements, and have included element uptake and toxicity data through logistic mathematical equations. So it also makes use of the incorporation of parameter information, which relates back to being an open model, that as soon as data is available in literature research or databases, it can be incorporated. But I, of course, also use the data from individual bioassays, like from Edith and Marion, even maybe Isidora's, but also my own COSM experiments. So what are COSM experiments? They are indoor artificial simplified ecosystems that are used to simulate and predict the effect of natural ecosystems. So they're small aquaria that I put up in the lab and are supposed to represent the natural system. However, there's a wide variety within COSM experiments. They can be terrestrial, but they can also be aquatic. They can be super tiny and they can be super big. So the COSMs I use for my experiments look like this. This is a 200 milliliter cup. If you ever go to Starbucks, you might recognize it, but then there's coffee in it. I don't think you want to drink rare elements. It has a sediment layer with a water layer, and then I add some rare elements, and then I add only algae and dephnids. This is already quite complex because you have the interactions of the rare elements with the sediment and the water, and then also with the 2 different species, and the species interact. But I can make it even more complex. So the next step is having a 2-liter microcosm, where, as you can see, I have up to 5 species, and I can even add abiotic factors like sediment resuspension. But I want to make my life even more difficult. So for one of my segments, I will go to the University of Lorraine together with where I will work under Laura Girandini, where I have these 20-liter mesocosms, where I have even more species, more abiotic factors, more complications. And with each step we take in this COSM experiment, the complexity increases, but also the variability, because more stuff is happening is more variability. There's another key challenge that I have with my model is that experiments are required to recalculate the relevant rate constants and the representation of chemistry of the rare elements. With other words, speciation. I think from all the talks we have heard today and in previous summer schools and workshops, we know how complex that can be. So my current model contains a predator-prey relationship, the reproduction rate, and the death rate, which is EC50, for all individual species. It also has a submodel for nutrients, for nitrogen and phosphate. And it has a so-called submodel growth limiting function, which is linked to this nutrient submodel. And in order to get to link it to the controlled conditions we use in the experiment, I have added a temperature function and a light function. So this is my current food web model, and don't panic, I will walk you guys through it. As you can see, we have the different species, all in different colors. Sorry for people who are colorblind. And we have these kind of dotted lines that are an influence. So you have to tell the model X is related to I. But for these fully colored arrows, the model itself already formulates a mathematical equation, and I can just adjust it and change it. You see that some of these boxes are single lined and others are double lined. Well, these double line boxes, there's something behind that. It's either a submodel or an Excel table. So let's focus on the algae population. So if I click on this box, this is the formula that shows up. As you can see, it's the compartment for the algae population. So I always know in which compartment I'm working with. And you will see that formula starts or is identified as the algae population divided over time. For this, we look at the natural growth rate of algae times the reproduction rate of the EC50 caused by the rare earth elements. The rare earth elements are here given in dose. For this current setup, it's set at 0. So for now, no rare earth elements, very sad. But you can find this in my parameters, which is a double box, which leads to Excel table where I put in all the information I get from literature research and databases. Then the next part is the algae population divided by the carrying capacity. For those who are not familiar with ecology, carrying capacity is how much an ecosystem can handle. So if we have 100 animals, or the ecosystem has the ability to feed 100 animals, and one of the animals get a baby, we have 101 animals. That's too much for the ecosystem. The ecosystem will collapse, and your population is done for. So that's what this carrying capacity takes into account. Then, as you might remember, algae get eaten by definite. So for the algae population, that's also something you need to consider when formulating your formula. So that's what we see here. We see algae minus the predation of algae to Daphnia. Then, as you might know, animals have the tendency, or organisms have the tendency to die on its own. So you also need to add a death flux, which is the natural rate of how much algae die off. So it's the algae population minus the predation minus the death flux. As you might see, I also take off the GLF, which is the growth limiting function I talked about before. And that can be found in this submodel. As you might see, there's no direct arrow pointing to the model itself. It is slightly linked to the dashed arrows, but that's okay because in a model maker tree, you have something that's called universal setting. And this universal setting, as soon as I click it, it becomes available in the whole model. So I don't need to draw extra arrows, which makes it a bit more clear to the eyes. So when I double click on this function, this is what shows up. So this time function and photo period is what I talked before. These are the controlled conditions of our experiments. And then for each species, we have a temperature function, a light function, and the nutrients, nitrogen and phosphate. And if everything goes well, then this happens. Your population starts to grow. If it doesn't go well, of course, if not the optimal functions for the growth limiting function are reached, then it actually decreases. So basically, I won't talk you through this whole difficult mathematical equation. And if you do it well, you get a result that looks like this. So this is the primary result. And as you see, these are the algae. So we have a slow increase, then stabilizes, and then it kind of shows it Lotka-Volterra equation or graph. This is, however, a bit concerning because when we look at the ostracods, you see it does it in the beginning and then it's completely stable. If you know anything about the natural system, is that it's never stable. So this shows that there's an error in, or a lack of data in my model. I told you I did not add any rare elements, so there's definitely something going on there. When I looked further into that, I was like, I need to check what did I include and what did I not include. So I included the predator-prey relationship. I included the reproduction rate. I included the death rate. I included the submodel growth limiting function, but the nutrient submodel was under construction. So the nutrient submodel can be found here. As you can see, all the species lead to the nutrient model, and then there's an outflow that through the dashed lines go into the growth limiting function. When we click on this box, we will see this. Basically, when you think of animals, they die off, they fall onto the ground and get decomposed and then turn in some way back to the organic nitrogen, and then there's some other functions, and it should be a cycle. It's a nutrient cycle, basically. The problem is that because I'm still constructing this, my model keeps crashing, and I have difficulties trying to figure out what the possible issue could be. What I suspect is that the amount of nutrients that go into the nutrient cycle is not enough to sustain the uptake of the individual organisms. So further research is required, which I do through the mesocosm or the cosm experiments. Right. So it's not bad that my model crashes because you are only a real modeler when you hit your first crash. So when I got my first crash, I called Jordi and I was like, yes, it happened. So we had a small party, very nice. Also leads to learning some lessons. So I learned that extensive literature research is really required to get the correct parameters. Sometimes the research is, the studies that have been done are lacking, and I need to perform my own studies through cosm experiments. And those cosm experiments are also required for gathering information on toxicity of rare elements in the food web, because there are a lot of interactions happening again, of which most studies only focus on individual bioassays. So we miss a great part of the knowledge I need to know. I also learned that a deep understanding is necessary of all processes involved in the food web. As I said before, modeling is multidisciplinary. Some of these are now, I struggle a lot with chemistry. That shows up in my model. And also food web modeling is necessary to get a deeper understanding of the natural system. So it's all linked together. Also, it's important that you're not too ambitious when you model, because ambition leads to complexity. And what's important is that when you model is that you have find the balance between simple and complex. So you start very simple and you work your way to the way more complex situations until you can't go further. Does this mean that I will now stop after my beautiful crash with the nutrients? No, because it's still a necessary thing to understand what happens in our model. It just might mean that I make concessions by saying, oh, I will cut out this species, or the precipitation is too much for me to handle at the moment to model, so I will not include it. It's making choices. And that's all what modeling is about, making choices. So what's next? Well, as I said, I lack some knowledge. So the first next step is that I will continue performing my microcosm studies and my mesocosm studies, and also try to see which bioassays I can still perform within the time of my PhD. But also for the model itself, I want to integrate the bentopelagic coupling for lanthanum and gallium, which is this sedimentary suspension. So when I suspend the sediment, how much of the rare elements end back up in the water. Also, as I might have said, the current model is based on acute toxicity and not on chronic toxicity due to the time limit of the model. So that's also something I need to figure out. And also, I want to integrate a reparation mechanism model, which considers that healthy organisms can get into your ecosystem or in your ecosystem, but that sick animals also can get healthy again. And then also what I personally think is important is considering the precipitation of lanthanum and gallium, which I currently have based on Geochemist Workbench, but Marion and I are working on modeling with Mintech in the algae. And I know some of you are more knowledgeable with Frixie. So I have to see what I do with it. And then at the very end, when I'm satisfied with my model, it also needs to be parameterized, calibrated, validated, and verified in order to be able to publish it. So basically, I start here, and then I can use the data to incorporate in my model. My model can then tell me a bit more about the natural system, for example, our beautiful river Elbe. As I said, it's a tool to understand the natural system. It's not a replacement of the natural system, but it can help us with policymakers so that it's easier and more justifiable to understand what's going on. And it also helps us understanding where are the data gaps, which studies do we still need to perform. And yeah, thank you. If there are any questions.
Thank you, Chantal. Do we have any questions?
Everyone's so bored they fell asleep.
No, I would have a question actually, because you compared, I think you said it also at the end, but I might have missed something. You said that you do everything according to EC50, which is something calculated for acute toxicity. And why then, knowing that it's for chronic, we would still have lower concentration? Why the EC50 and not the EC10 or even the NOEC or something like this?
Because from literature research, I've known that in recent years, a lot of people start to move away from the EC10 and the EC20 because they're close to the NOAC. And because of most data that we have available right now is for the EC50. So it's more like a personal choice than a necessary requirement. And I can still change it at any moment in time because it's an open model. So if I later on decide, well, those EC50s were not a smart idea, I can just move on to the EC10s.
And because if you EC50 times 30, I think it was.
Yes. So
why this value?
So in order to make my model work, here. So in order to make my model work, I need to add a half-life time for the gadolinium. So I tried a few runs where I added, instead of saying like, oh, the dose for gadolinium or lanthanum is 0, I added 100 milligrams per liter in the system just to see what would happen. And then in order to let my model run, I need to add a half-life time of the gadolinium because it kind of participates and moves away in the system. It flows out. That's what happens in the natural system. And so I kind of had to come up with a number, and Jordi and I were just like, let's do 30. That's where this 30 comes from. Yes.
Okay.
Yes.
Which would be in day, like what would be the units of this 30?
In days. But as I said, the 30 is just a made up. Just I added there to try to make it run, and I copied this literally from the last run I did. So that's why it's still in there.
Okay.
I do have one question because I think there is something I misunderstood. You mentioned that models, so that's how I understood it. Mm-hmm. Models have a validity of 14 days.
Okay, no, not all models have, because for example, the ICP-CC, which we use for climate modeling, those of course is in several years. It's just for this current setup with the food web model, because most of the data that's available is acute. we decide to optimize it to 14, and I can extend it. It's just that I'm not at the point where I can extend it yet. So to see how good my model runs, we decide to go for 14.
Okay. Is the goal to extend it or 14?
Yes, it's the goal to extend it. That's also why I said, like, I want to change the EC50s of the acute test to the chronic test, because chronic tests are longer and our natural systems are not there for 7 or 14 days. So it's not that they suddenly disappear. So yeah, but as I said, it's a prototype. I still need to build on it a lot, still need to play with it a lot. So. Yes.
Thank you.
So you can change the dates, right? You decide everything about the model.
Yes. If I suddenly want to add, I don't know, trees, I can do that.
Okay.
It's just how complex is it?
Yes.
Yeah, you show the figure with a line or a curve for each species.
Mhm.
I didn't understand what those they represent.
So it's the population dynamics over time. So each line basically shows how much a population grows, and then
Like abundance of
Yes.
Okay.
yes,
And the time, it's in days, then in that case, in hours, and what is it?
yeah, it's here it's in hours, but this is not the 14 one. This is so the problem is, as soon as your model crashes, it doesn't do anything anymore. It doesn't give me any results. So this is also an old result before I did my nutrient model. As you hear, there's a lot of issues.
I
Oh.
dare to make an extra scientific but linguistic note, which is devoted not to an individual, an esteemed colleague, but can be of some utility for other colleagues. For the element LA, lanthanum, it's compulsory to push the accent on the first A, and one should not forget that the T is a theta, which is pronounced th with a th in English. And so the correct pronunciation that you can check on Google Translator is lanthanum and not lanthanum. And second, for the same reasons that I placed in my short talk, let's remember that in Latin, in classical Latin, the sound ñ does not exist, as it does not exist in German, for instance. And so Daphnia is magna and not Daphnia magna. Because if we Neo-Latins have this sound, it doesn't exist in so many languages and not in classical Latin. So Daphnia magna and lanthanum. Excuse me for the extra scientific point, but it was not a personal confidence with the esteemed colleague who needed this, this to be precise.
Thank you, Joel.
Beg your pardon.
Yeah. To just come back on the presentation. With your model, are you able to plot the relationships between species? Or?
Yes, so that's one of the ideas behind the model, that you can plot, or basically that I can model how species interact and what their relationships are and how they are affected mathematically. But as you might have heard, I still need a lot of work to do so.
Do we have any other questions, or should we call it today? Last chance? All right. Well, thank you very much, Chantal, for your time.